stem cell factors in lung development, and the nitrofen rat model of congenital diaphragmatic hernia. So CDH is a developmental disorder that affects about 1 in 3,000 newborns, and the people affected with this disease, they have a hole in the diaphragm through which the abdominal organs herniate, and they also have hypoplastic or underdeveloped lungs. Now the mortality rate for this disorder is still very high, about 40 to 62%, despite the fact that repairing the hernia itself is actually becoming relatively easy and with less and less invasive procedures. So we're wondering what's going on there. So this is a photo that shows classic CDH, where we have the liver herniating on the left side of the body and moving the lung up and out of position and restricting its space to grow. But in CDH, there isn't much evidence or any really good evidence as to a genetic cause. So it leads us to think that there might be some kind of epigenetic link causing this disorder. So that leads us into microRNAs. The microRNAs are short 22 nucleotide RNAs which bind to mRNAs and inhibit translation. So this is an image of a microarray performed previously by Dr. Kaiser comparing control and CDH in humans. And we have highlighted here 2 different microRNAs which are both vastly overexpressed in the CDH. We have MIR200B and MIR10A. For the purposes of my work, we focused on the MIR200B, because it had already been started working on in the lab. So MIR200B is typically thought of being an epithelial phenotype enforcer for a microRNA. The reason for this is that this E-coherent is a protein that's a marker of the epithelial phenotype, and it's repressed by ZEB. ZEB is repressed by MIR200B. So through a double negative, MIR200B enforces the phenotype by allowing the expression of the E-coherent. So knowing this, in situ hybridization was performed. It's basically staining, but for microRNAs, for MIR200B. The dark blue is MIR200B, and these spots here are where MIR200B is overexpressed. What's interesting here is that these are actually the tips of airways where we're seeing a lot of expression, and this is in day 15 lung. So the lung is still growing and still branching, but why do I care so much that it's in the tip compared to other regions? Well, it's because in the tips, it's already known in lung development that the tip cells act sort of like stem cells. They grow and they branch and they have the capability to produce all other lineages in the lung. So why would something that is thought to be an epithelial phenotype marker, something that's supposed to be endpoint, be showing up in these cells that aren't fully developed yet, that are leaving behind other things? So the thought was maybe it's changing those into epithelials through downregulating other things. So many different options are coming up. So we started looking into different stem cell markers which could be related to MIR200B. The first one that came up was SOX2. SOX2 is a well-known target of MIR200B, and it's also well-known for its ability to induce pluripotent stem cells. But recently, what came up was that SOX2 is actually involved in a negative feedback loop with MIR200B, shown just earlier this year by Wang et al. So we started looking for what else could SOX2 be done. Is SOX2 known in lung development or not? What we came up with was that SOX2 is actually a known proximal marker, so not the tip, but it's actually known to be in the center. So we were wondering, okay, well, what if it was mis-expressed? So we found a study where they had an overexpression of SOX2, and when SOX2 was overexpressed in this study in the tip region, it impaired the branching development. So then we're thinking, well, what if in the nitrofen model you have an overexpression of SOX2 in the tips, and then you have an overexpression of MIR200B to compensate, but the branching damage has already been done, and that's why we're seeing some hypoplasia. So that was the hypothesis we went forward with. That a normal expression of SOX2 into the distal region in the nitrofen rats caused impaired branching and led partially to the hypoplasia that's demonstrated in the CDH model, this CDH model. So going forward, the methods we used was nitrofen rat. I've mentioned it a few times now, but I haven't explained it. Nitrofen is a herbicide, but when you administer it to pregnant rats on a very specific day, at day 9 of gestation, it causes a CDH model, whereby 100% of the pups have hypoplastic lungs, and 60 to 90% develop hernias as well. So using this model and immunohistochemistry, because we're looking for localization, we were looking for localization at the start, we weren't looking for concentrations or anything like that. We're just trying to see, is it expressing in the tips in the nitrofen or more? So we went forward with that. So this is our E15. On the left we have control, and on the right we have nitrogen. The brown staining is positive staining for SOX 2. So what we'd be looking for here is we were hoping to see some kind of staining in these smaller airways. But when we look at these, we just see staining in these large airways in both cases, control and nitrogen. And when we increase the magnification, it doesn't, you still can see that it's very clearly just in these epithelial cells of these airways. So it's not spreading into the mesenchyme, and it's only in these large airways here. It's not spreading out to the smaller ones around them. So we checked with E18, a later day point, just to confirm our findings. And again, we're only seeing it in epithelial cells in large airways. There's no difference. There isn't a difference between control and nitrogen. So that's the conclusion that we drew, was that the proximal distal patterning of SOX2 is not changed in the nitrogen model based on the immunohistochemistry we performed. So after SOX2, I moved on to another protein, nanog. Nanog is a stem cell marker. It's famous for being able to maintain pluripotency of embryonic stem cells and induce pluripotent stem cells. So we wanted to look at it and see if it had some kind of role, if it was being expressed in these tips, because no one had published before. So it was suspicious that no one had done it, but we were curious to see what would happen. So we started again with immunohistochemistry, but then based on our immunohistochemistry this time, we advanced forward to Western blotting to compare between different gestational time points and different between control and nitrogen. So when we start this E15, there isn't any specific staining in either control or nitrogen in immunohistochemistry. It's almost blank. But then when we move forward to the E18, this brown here again in the airways and in the mesenchymal tissue this time, not just like the SOX2 where it's only in the epithelial, we see specific staining. But we don't see staining in these smooth muscle cells because they're derived from a different origin. And then when we go forward to E21 again, we have the same pattern as E18. But looking at the IHC, we couldn't see any differences between control or nitrogen. So we went forward to Western blotting to confirm this change in expression. But what we got was actually a slightly different result, but still very interesting for us, was because now in the Western blot we can see expression in the E15. There's two isoforms of nanog, so you have two bands very close together here. Check that that's the right band width and that's the right size. But what we do notice here too is that this E18 nitrogen is much greater than this E18 control. And we saw that this trend in 3 different plots that I ran so far, where the nitrogen E18 seems to be higher than control, but the rest of these agree. So I have 3 graphs here where I've quantified. This is control, nanog expression in control animals throughout development. We use E15 as our cross-reference between the different plots, setting it to 1. And we show that between E15 and E18, interestingly, on the Western blots in control, there doesn't appear to be difference, which is confusing since it doesn't agree with the IHC, but this trend was the same in all 3 of our Westerns, so going forward with it. But the E21 here is much more expressed than in any of the days. Statistically relevant using one-way ANOVA and paired T tests for both of the other days, it's much more. This could be just a developmental change as the lungs change shape, they express more. But the interesting fact was that in the nitrogen, there's a much different pattern. The trend is different. It doesn't just accelerate at E21, there's a spike at E18. Although right now, because of the variation, there isn't a statistical difference, the trend between all the different plots is the same. We have an increase in E18 and then back down a little bit in E21. So we compared just a quick graph to show the E18 control versus nitrogen. So using E18 control as the reference point between the different plots and then nitrogen compared. We showed this, and again, right now there's too much variation for it to be statistically relevant, but it's because the sample size is only 3. So once we increase it and we start to spread it out a little bit more, this could be an interesting thing. So seeing this wasn't expected based on what we were doing. So I've been doing some literature search and trying to find out what could nanog be involved in. And what I found was that there's a signaling pathway using bone morphogenic protein 4, and it's essential during branching of the airways. But nanog has also been shown to be capable of interfering with that signaling through SMAD1. So if there's something going on here, it could be why nanog could have an effect on the branching that we're because the branching is impaired in the nitrogen model. So this is an essential pathway, and if NANOG is interfering with it in this, it could be an explanation as to why we're seeing a difference between nitrogen and control. If it goes up in nitrogen, all of a sudden the branching is impaired improperly too early at 18 instead of at 21. So the future studies would be to increase the trials and focus on the E18 so we can get some statistically difference between nitrogen and control, confirm that it's there, the trend that we're seeing. And then after that, we have primary cells from E18 rats that have been isolated and immortalized in the lab already. So we can alter the expression of NANOG in those cells using inhibitors or using plasmids to overexpress. We could then we could then check the effect on the BMP signaling proteins and see what's happening downstream, if it is in fact changing what's going on. So just quick summary, the CDH is 1 in 3,000, and most of the mortality is due to the hypoplastic lungs. And hypoplastic lungs are seen because of this impaired branching. So the more we can understand about this impaired branching, the better we will be able to understand the disease in general. So I'd just like to thank Dr. Kaiser for having me in the lab, Manitoba Institute of Child Health, Tim Hortons for the donation of the studentships this summer, and everyone else who's funding the grants.
I asked them all my questions. Okay.
Theo, so obviously moms aren't exposed to nitrogen, right? Like human moms.
Yeah.
So how do you know that it's not just the toxin that does all of this crazy stuff with your DNA, and it's not and it has something to do with the actual?
Yeah, we're not exactly sure what's going on. The nitrogen model isn't fully understood yet. There is some evidence that it's interfering with the retinoic acid pathway, because when you treat with nitrogen and then you treat with retinoic acid, you can reverse the effect and get back to normal pops kind of thing. But in humans, we can't. There's no studies on what type of consumption there is, or you can't do that type of thing. So we're not really sure.
Can we get exposed to nitrogen? Like, is nitrogen an environmental?
No, it's a herbicide that's
Not used anymore?
Not used anymore.
Was in the 60s and 70s.
So were there more congenital diaphragmatic in the 60s and 70s? No.
But this is the high dose. Would be like kilos of nitrogen.
Be like lying in the field.
Swimming in the nitrogen. Yeah. But like what he was saying with retinoic acid is true. If you measure retinoic acid blood levels in babies and mothers, babies have lower levels of retinoic acid too. So there is a clear link there, but we don't really understand it.
That's excellent, thank you.
Anybody else? Okay, let's move on to